So, me caveman, let's go. Number 10. Cheddar Man's Reconstruction. A fine drill bites into a 10,000-year-old skull. The dust it lifts is not from the cranium or the teeth. It comes from the petrous bone, the dense knot of bone that wraps the inner ear. This is the single best vault for ancient DNA in the entire human body, and inside this fragment sits the genetic code of the oldest near-complete modern human ever found in Britain. His skeleton was recovered in 1903, deep inside Gough's Cave in Cheddar Gorge in Somerset. He lived about 10,000 years ago in the early Mesolithic, roughly 300 generations before anyone alive today. In 2018, a team led by Tom Booth, Selena Brace, Ian Barnes, and Johan Diekman pulled a high-quality genome from that inner ear bone. The result reframed what Britain's first people looked like. The most likely profile was dark to black skin, dark brown or black curly hair, and blue or blue-green eyes. That combination came as a shock because it inverted a long-held assumption. Many people had assumed pale skin came first in Europe, and light eyes arrived later. Cheddar Man suggested the opposite order. He carried light eyes while still carrying dark skin. The genetic reason is specific. The pigment lightening gene variants that dominate modern Europeans, named SLC24A5 and SLC45A2, were largely absent from his genome. Those variants spread widely only later, arriving with Neolithic farmers from Anatolia. Cheddar Man belonged to an earlier group that geneticists call the Western hunter-gatherers, and he was not alone in this. Mesolithic individuals from La Bretagne in Spain, from Lochbourg in Luxembourg, and from sites in Hungary showed the same dark skin signal. In that context, his pigmentation was ordinary, not exotic. The reconstruction made it real. The Dutch paleo artists Alphonse and Adrie Kennis built a face from a three-dimensional printed scan of the skull, combined with the genetic pigment data. A Channel 4 documentary aired the result in February 2018, and the image traveled the world. Then came the backlash. The Daily Mail asked whether Cheddar Man was white after all. The dispute exposed a real scientific limit. Susan Walsh, who helped develop the pigment prediction system, noted that confidence on skin tone sat near 76%, not certainty. Booth himself later admitted the team had perhaps overstated how sure they were. The prediction tools are trained mostly on living Europeans, so reaching 10,000 years back stretches them. That tension between a probabilistic genome and a public hungry for a definite answer is exactly where these stories ignite. Thousands of miles east, another set of faces was about to break a different theory. Number 9. The Tarim Basin Mummies. In the Taklamakan Desert of Xinjiang in northwest China, the dead do not rot. The air is bone dry and the soil is heavy with salt, so bodies dry into natural mummies with eyelashes and hair intact. The oldest of them, from the Xiaohe and Gumugu cemeteries, date to roughly 4,100 to 3,700 years ago. What made them famous was their appearance. Many had high noses, deep-set eyes, and fair or reddish hair. They were buried in woven wool with tartan-like patterns and tall felt hats. Two of them, called the Beauty of Xiaohe and the Beauty of Lulan, became icons. For decades, those Western-looking faces fueled a confident theory. Scholars argued the mummies were Indo-European migrants, perhaps Tocharians, or distant relatives of European peoples, who had trekked thousands of miles east across Eurasia. It was a tidy story, and it seemed obvious from the bodies alone. Then the genome arrived. In October 2021, a team led by Christina Warrener, Chungwen Zhang, and Yinkyu Kui published a study in the journal Nature. They sequenced the genomes of 13 of the oldest Tarim mummies, along with 5 individuals from the nearby Dzungarian Basin. The result demolished the migration theory. The Tarim mummies carried no Western steppe ancestry at all. They were not migrants from the West, and they were not newcomers of any kind. Instead, They were direct descendants of a population called the Ancient North Eurasians. These were Ice Age hunter-gatherers who had once ranged widely and then mostly vanished. The early Tarim people carried about 72% of this ancient North Eurasian ancestry, plus around 28% from ancient Northeast Asians. To put that in modern terms, this old lineage survives only in fragments today. The highest proportions, around 40%, appear in indigenous Siberians and Native Americans. The Tarim people were, in effect, a living relic, a Pleistocene population that had survived in isolation into the Bronze Age. Yet genetic isolation did not mean cultural isolation. They farmed wheat, millet, and barley, herded cattle, and made a kefir-style fermented dairy. They buried their dead in boat-shaped coffins beneath towering wooden posts, a mortuary style with no clear parallel anywhere. The lesson Warrener stressed is one that runs through this whole list. Genes, language, and culture do not have to move together. A people can look one way, speak another influence, and descend from a third source entirely. The finding also carried modern weight, because the history of Xinjiang is fiercely contested. The DNA undercut the old claim of early European presence, and it equally undercut the opposing claim that the Mummies were Han settlers. They were neither. From a contested desert, the trail now leads to the banks of the Nile. Number 8. The Abusir el-Meleq Egyptian mummy genomes. For years, researchers believed ancient Egyptian DNA was a lost cause. Egypt is hot, and heat shreds genetic material. The embalming process, with its resins and high temperatures, was thought to finish the job. So, when a team led by Verena Schooneman and Johannes Krauss announced the first reliable genome-wide nuclear data from Egyptian mummies in 2017, it was a technical landmark on its own. The site was Abusir el-Meleq in Middle Egypt, about 100 kilometers south of Cairo. The team sampled 151 mummies. They recovered mitochondrial genomes from 90 of them and full nuclear genome data from 3. The samples spanned a wide window from the late New Kingdom through the Ptolemaic and Roman periods, roughly 1400 years before the Common Era to 400 years after it. The headline finding stepped straight into one of history's most charged debates. The ancient Egyptians of this site were genetically closest to Near Eastern and Levantine populations. They resembled the peoples of the ancient Levant, Anatolia, and the Neolithic Near East. And in a detail that drew the most heat, they carried less sub-Saharan African ancestry than living Egyptians do today. The study found that modern Egyptians carry about 8% more sub-Saharan African ancestry than the Abusir el-Meleq individuals. That gene flow from the south appears to have increased mostly within the last 2,000 years. That single result reignited a centuries-old, deeply political dispute over the ancestry of the ancient Egyptians. Afrocentric scholars had long argued that ancient Egypt was a black African civilization, and some read the Near East affinity as a challenge to that view. Other researchers pushed back with an important caveat. Abusir el-Meleq is a single site in the north in Lower Egypt. It may not represent Upper Egypt, far to the south, or Nubia, both of which had stronger African connections. One northern village cannot speak for a civilization that stretched 1,000 miles along the river. There was another striking pattern in the data, separate from the politics. The mummies showed remarkable genetic continuity across 1,300 years, even as Persians, Greeks, and Romans conquered the land in turn. Rulers changed, but the core ancestry of the people held steady. The team reached this through a method called hybridization capture, which fishes out genuine ancient DNA from heavily damaged and contaminated samples. They deliberately avoided racial labels in the paper. The media did not. Coverage mapped the result straight onto modern identity at once, and the genomes became a fixture in arguments over the ethnicity of the pharaohs. From Egypt, the controversy travels to a single skeleton in India that lit a national fire. Number 7. The Rakhigarhi Indus Valley Genome. Rakhigarhi, in the Indian state of Haryana, is the largest known settlement of the Indus Valley civilization, which flourished from about 2600 to 1900 years before the Common Era. In 2019, a study in the journal Cell, led by Vasant Shinde and Neeraj Rai, with collaboration from David Reich's Harvard Laboratory, reported ancient DNA from this Harappan city. The numbers tell you how hard the work was. The team sampled 61 skeletons. Only one yielded enough authentic DNA to sequence, an individual dated to around 2,500 years before the Common Era. India's hot, humid climate destroys DNA fast, so the researchers screened many bones and processed the petrous bone in a dedicated clean laboratory to win even that single genome. The result was a mix. The Rakhigarhi individual carried Iranian-related ancestry, blended with ancient ancestral South Indian hunter-gatherer ancestry. And then came the detail that turned a quiet paper into a political event. The genome contained 0 Central Asian steppe ancestry. That matters because steppe ancestry, derived from the Yamnaya herders, is the genetic marker tied to the later arrival of Indo-European, or so-called Aryan peoples. Finding none of it in a Harappan more than 4,000 years old was explosive. The Iranian-related component, moreover, appeared to have branched off before the rise of farming, which suggested that agriculture in South Asia was a local development rather than an imported one. The reaction split along ideological lines almost instantly. Hindu nationalist commentators seized on the absence of steppe ancestry as proof that the Aryan migration never happened, and that the Vedic people were indigenous. Mainstream geneticists answered that the result was fully compatible with later migrations. A companion paper, published the same week by Narasimhan and colleagues in the journal Science, showed that steppe ancestry did enter South Asia, but only after about 2,000 years before the Common Era, after the Indus cities declined. The Rakhigarhi individual simply lived before that arrival. David Reich himself stated that the data did not support an unbroken indigenous Aryan lineage. The single sample limitation sat at the center of the storm. One genome cannot capture the diversity of a civilization that numbered in the millions. Publication was reportedly delayed for years amid disputes over interpretation and political pressure. Vasant Shinde framed the broader meaning generously, saying that all of us are descendants of the Haruppans. Confidence in the result was strengthened by a match with 11 other Indus periphery samples found in Iran and Turkmenistan. Still, the same data fed opposite political narratives, and one ancient skeleton became a weapon in a living culture war. The trail now climbs onto the grasslands where the steppe story begins. Number 6. The Yamnaya steppe migration and the British replacement. The Yamnaya were pastoralist herders from the Pontic-Caspian steppe, the vast grasslands of modern Ukraine and southern Russia. They flourished from about 3,300 to 2,600 years before the Common Era. Their advantage was mobility. They had domesticated horses and wheeled wagons, and a herding economy that let them spread across open country at remarkable speed. Ancient DNA work by David Reich, Eske Villerslev, Wolfgang Hack, and others, from 2015 onward, traced Yamnaya-derived ancestry across an astonishing range, from Ireland in the west to Mongolia in the east, and India in the south. That spread is now the leading genetic explanation for how the Indo-European language family came to dominate so much of the world. The most jarring single finding came from Britain. In 2018, a study led by Inigo Olalde, published in Nature, examined the bell beaker phenomenon, a culture that swept across Western Europe in the Copper and Bronze Ages. The beaker people who reached Britain after about 4,500 years ago carried strong steppe ancestry, and when they arrived, the existing population was almost erased. The study estimated that roughly 90%, and possibly up to 100%, of Britain's Neolithic gene pool was replaced within a few centuries. The people who built Stonehenge were, in genetic terms, nearly wiped from the record, and replaced by newcomers. The scale of the dataset gave the claim weight. The 2018 Beaker study analyzed around 400 ancient genomes, one of the largest such collections assembled at that time. A replacement that fast and that thorough implied large-scale migration, demographic collapse, or both, rather than a simple spread of pottery styles and ideas. The steppe newcomers also carried specific genetic baggage. They brought variants for lactase persistence, the ability to digest milk as adults, and for greater height, traits that then spread across Europe. This is where the science became genuinely uncomfortable to publish. A narrative of 90% replacement is easily twisted. Far-right and white nationalist groups co-opted it into stories about conquest and blood. David Reich popularized these findings in his 2018 book, and that same year he published an opinion piece in the New York Times about genetics and race that drew an open letter of objection from 67 scientists and scholars. Reich has argued repeatedly that his work actually refutes racial purity myths, because every population turns out to be deeply mixed and constantly moving. Archaeologists also cautioned against the loaded word replacement, noting that sparse samples may overstate the case, and warning against projecting modern ideas of genocide onto prehistory without direct evidence. A battle site at Tallinsa Valley from around 1250 years before the Common Era shows Bronze Age Europe could mobilize organized violence, but motive across centuries is hard to read. From recent prehistory, the story now plunges far deeper into the human past. Number 5. Neanderthal Interbreeding. For more than a century, Neanderthals were treated as an evolutionary dead end. They were the cousins who lost, a separate species that vanished without leaving a trace in us. Svante Pääbo, working at the Max Planck Institute for Evolutionary Anthropology in Leipzig, set out to test that assumption by reading Neanderthal DNA directly. The early attempts, from 1997 onward, looked only at mitochondrial DNA, and they found no sign of interbreeding. That reinforced the clean replacement model, in which modern humans simply outcompeted the Neanderthals and moved on. The picture broke open in May 2010. Pääbo's team published a draft of the full Neanderthal nuclear genome in the journal Science. It was assembled mainly from 3 bones, between 38,000 and 44,000 years old, recovered from Vindija Cave in Croatia. The result was a bombshell. Every living person of non-African descent carries roughly 1 to 4% Neanderthal DNA. That meant modern humans and Neanderthals had interbred after our ancestors left Africa, somewhere around 50,000 to 60,000 years ago. The earlier studies had missed it for a simple reason. Mitochondrial DNA traces only the direct maternal line, a single thread, while the interbreeding signal was scattered across the rest of the genome. The central objection was contamination. Ancient bones are soaked in modern human DNA from everyone who has handled them, plus a swamp of microbial DNA. To answer that, Pääbo's team built rigorous cleanroom protocols, used bleach treatments, and developed statistical methods to separate genuine Neanderthal sequences from modern contamination. They relied on next-generation sequencing, which can read billions of short, broken DNA fragments at once. Skeptics offered an alternative, arguing that shared variants might reflect ancient population structure within Africa, rather than interbreeding. Later work largely refuted that explanation, and the interbreeding model held. The consequences ran deep, in more ways than one. Pääbo was awarded the Nobel Prize in Physiology or Medicine in 2022 for effectively founding the field of paleogenomics. Neanderthal inherited variants turned out to shape modern immunity, skin and hair traits, and metabolism. One Neanderthal-derived haplotype was later linked to a higher risk of severe outcomes from COVID-19. The finding was also sensitive in a way that intersects with race, because different human populations carry slightly different amounts of archaic ancestry. East Asians, on average, carry somewhat more Neanderthal ancestry than Europeans, which frustrates any simple narrative. The same laboratory went further that year, identifying an entirely new human group, the Denisovans, from a single finger bone in a Siberian cave, using DNA alone, with no diagnostic skeleton to guide them. The boundary between us and them had dissolved, and the next discovery would push DNA back to an age no one thought possible. Number 4. The Sima de los Huesos, 400,000-year DNA. Deep inside the Atapuerca cave system of northern Spain lies a chamber called Sima de los Huesos, the pit of the bones. To reach it, you crawl through narrow passages and then descend a vertical shaft about 13 meters deep. At the bottom sits the richest collection of Middle Pleistocene human fossils on Earth, the remains of at least 28 individuals. They lived roughly 400,000 to 430,000 years ago, and their skulls and teeth carry Neanderthal-like features. Most researchers class them as early Neanderthals, or as Homo heidelbergensis, the ancestral stock that gave rise to them. In December 2013, Matthias Meyer and Svante Pääbo's team published a near-complete mitochondrial genome from one of these Sima bones in the journal Nature. At about 400,000 years old, it was by far the oldest hominin DNA ever sequenced. It shattered the previous record, which had belonged to a Denisovan only around 80,000 years old. The achievement alone was staggering, but the result was stranger still. The Sima mitochondrial DNA was more closely related to Denisovans than to Neanderthals. That made no sense at first glance, because the skeletons looked Neanderthal, while the genetic signal pointed east, toward the Denisovan lineage that ranged across Asia. The mismatch was so unexpected that the team withheld the result and rechecked it again and again, afraid that contamination or error had fooled them. The technical feat behind it was extraordinary. They extracted DNA from just 2 grams of bone powder and used new methods built to capture extremely short, badly damaged fragments. The reason any DNA survived for 400 millennia comes down to the cave itself. The cold, stable conditions of Atapuerca preserved genetic material far longer than anyone had believed possible. The puzzle was resolved 3 years later. In 2016, the same team published nuclear DNA from the Sima hominins, also in Nature. The nuclear data told a clearer story. The Sima people were in fact on the Neanderthal lineage, more closely related to Neanderthals than to Denisovans, just as their bones suggested. The earlier mitochondrial mismatch was explained as an ancestral mitochondrial type that was later lost from the Neanderthal line, or as early gene flow that muddied the signal. The nuclear result also pushed the population split between Neanderthals and Denisovans to before 430,000 years ago. The deeper lesson endures. Anatomy and genetics can point in different directions, and the bones alone can mislead you. From the cold caves of Europe, the story leaps to a dry lake bed on the far side of the world. Number 3. Mungo Man, Lake Mungo 3. In 1974, the geologist Jim Bowler found a human skeleton eroding out of the ground at Lake Mungo in the Willandra Lakes region of New South Wales, Australia. The body, known as Lake Mungo 3, had been buried with care and sprinkled with red ochre, making it one of the earliest known ritual ochre burials anywhere in the world. Nearby lay Mungo Lady, found in 1968, one of the oldest known cremations on Earth. The dating of Mungo Man has been argued over for decades, with estimates ranging from about 30,000 to 62,000 years. A figure near 40,000 to 42,000 years is now widely accepted. Either way, he is among the oldest anatomically modern humans found outside Africa. In 2001, Greg Adcock, Alan Thorne, and colleagues published mitochondrial DNA from Lake Mungo 3 in the journal PNAS. They reported that he carried a mitochondrial lineage found in no living person, a line that did not descend from the common maternal ancestor known as mitochondrial Eve. Alan Thorne was a leading champion of the multi-regional model of human origins, which held that modern humans evolved in several regions at once rather than only in Africa. he used the Mungo result to challenge the dominant out-of-Africa theory head-on. If a 40,000-year-old Australian carried a lineage older than the African source, the tidy single-origin story might be wrong. The challenge collapsed under scrutiny. Critics, including Alan Cooper and Michael Hofreiter, pointed out that the team had not applied the standard authentication criteria that ancient DNA work demands. The reported sequence showed cytosine to thymine changes, exactly the kind of chemical damage that accumulates in old, degraded DNA after death. Recovering genuine DNA 40,000 years old from a hot Australian environment is extraordinarily difficult, which made post-mortem damage and contamination the far more likely explanation. The field has since broadly rejected the Mungo mitochondrial result, and the out-of-Africa model still stands. The scientific dispute was only half the controversy. When photographs of Lake Mungo 3 appeared in Australian newspapers in 2001, Aboriginal communities were angered because the remains had been studied and displayed without their consent. The skeleton had been held at the Australian National University under Alan Thorne's care for decades. Repatriation demands grew from the Pakanti, Nyampaya, and Muti Muti peoples. In 2015, the university formally handed the remains to Aboriginal elders and expressed sincere regret for their removal. In 2017, Mungo Man was returned to Lake Mungo in a repatriation ceremony. The case binds two controversies together, a discredited genetic claim and an unresolved fight over consent. That second thread leads straight to a lock of hair in a museum drawer. Number 2. The Aboriginal Hair Genome. In September 2011, a team led by Eski Willerslev published the first complete genome of an Aboriginal Australian in the journal Science. The source of that genome was not a blood sample or a fresh swab. It was a 100-year-old lock of hair, kept in a museum collection. The hair had been collected in the early 1920s by the British ethnologist Alfred Court Haddon, who obtained it from a young Aboriginal man, reportedly during a train journey from Sydney toward Perth. The donor came from the Goldfields region of southern Western Australia. Hair was a clever choice because it can yield endogenous DNA, the individual's own genetic material, with relatively low microbial contamination compared with bone in some conditions. The scientific result reached deep into human prehistory. The genome showed that Aboriginal Australians descend from an early, separate dispersal out of Africa, distinct from the migration that produced most Europeans and Asians. The study estimated this dispersal at roughly 62,000 to 75,000 years ago, earlier than the wave that gave rise to most other Eurasian peoples. That made Aboriginal Australians one of the oldest continuous populations living outside Africa, with extraordinarily deep roots on their continent. The findings supported a model of multiple dispersals out of Africa, rather than a single tidy wave, and it reinforced the archaeological evidence for very ancient occupation of Australia. The controversy was not about the science. It was about consent. The team had sequenced the entire genome of a named individual without first obtaining the agreement of his living descendants. That decision became a landmark in the debate over who owns ancient DNA. Willerslev argued that the original donor had given his hair willingly and that a Danish bioethics review board had approved the work. After publication, the team sought and received an endorsement from the Goldfields Land and Sea Council, the body representing the relevant Aboriginal community. But the sequence had come first, and the consultation second. That ordering set a new standard. The case helped establish the principle that community consultation should come before genomic research on ancestral material, not after it. It also shone a harsh light on the vast collections of hair and tissue held in Western museums, gathered during the colonial era, which now represent an enormous and ethically fraught archive of recoverable genomes. The hair sample had avoided disturbing a burial, yet it raised an entirely new set of questions about the colonial collections themselves. Willerslev went on to prioritize indigenous partnership in his later ancient DNA projects, a shift shaped in part by this episode. A single lock of hair had demonstrated both the scientific power and the ethical hazard of these archives in one paper. The final case carries that exact collision of science, ethics, and identity to its most explosive point on a riverbank in Washington state. Number 1. Kennewick Man, the Ancient One. In July 1996, two young men wading in the Columbia River near Kennewick in Washington state found a human skull. The skeleton that emerged was about 8,500 to 9,000 years old, one of the most complete early American skeletons ever recovered. The trouble began with its description. The anthropologist James Cheddar examined the skull and called its features Caucasoid. That single word set off a sensation. Some speculated the skeleton resembled Europeans, or Polynesians, or the Ainu people of Japan. The idea that an ancient American might not be ancestral to Native Americans was both scientifically provocative and politically charged. Under the Native American Graves Protection and Repatriation Act, passed in 1990, tribes of the Columbia Basin claimed the remains, whom they call the Ancient One, for reburial. A group of scientists sued for the right to study the skeleton first, arguing that its affiliation with any modern tribe was unproven. The legal battle ran for years. In 2004, a federal appeals court ruled that the remains were too old to be linked to any present-day tribe under the law and allowed scientific study to proceed. Throughout the fight, the skeleton was held at the Burke Museum in Seattle. It became the most famous repatriation case in the history of American archaeology, a direct collision between scientific access and indigenous sovereignty. DNA settled what skull shape could not. In June 2015, Eski Villerslev's team at the University of Copenhagen published a genome analysis in the journal Nature. They extracted DNA from a hand bone and compared it against genomic datasets from around the world, including the Ainu and Polynesians, the very groups the skeleton had supposedly resembled. The result was decisive. Kennewick Man is more closely related to modern Native Americans than to any other living population on Earth. The DNA showed a specific affinity with the Confederated Tribes of the Colville Reservation, who had taken part in the study. The genome flatly contradicted the earlier reading of the skull. The case became a lesson in the limits of craniometrics, the measurement of skull shape, and the clarity of genome-wide DNA. Appearance had pointed one way, and ancestry pointed firmly the other. After the genetic finding, legislation was folded into the WIIN Act, signed by President Obama in December 2016 to expedite the return of the remains. On February 18, 2017, the Ancient One was reburied by a coalition of Columbia Basin tribes at an undisclosed location. The two-decade struggle reshaped policy and practice across the United States on how ancestral remains may be studied, and on the central role of tribal consent. It stands as the defining example of a DNA discovery whose publication was delayed and contested for 20 years by the tangled forces of law, ethics, and identity. Across all 10 of these cases, a single thread keeps surfacing. Look back across the list, and one rupture appears again and again. The bones say one thing, and the genes say another. Cheddar Man's skeleton gave no hint of his pigmentation, and only DNA filled it in. The Tarim mummies looked like Western migrants, while their genomes revealed an Ice Age relic population of the East. The Abusir el-Meleq mummies were assumed by some to be one thing, and their nuclear DNA pointed toward the Near East. Kennewick Man's skull was read as Caucasoid, and his genome proved him a Native American ancestor. In every case, appearance misled, and the genetic data corrected the record. The strangest version of this rupture came from Sima de los Huesos, where the skeleton said Neanderthal, but the mitochondrial DNA said Denisovan, until nuclear data restored the agreement. The recurring loser in these stories is craniometrics, the old practice of reading ancestry from skull measurements. Time and again, that method delivered confident answers that genome-wide DNA later overturned. The deeper reason is that skull shape responds to climate, diet, and chance, while DNA records descent more directly. A few technical truths run underneath all of it. The petrous bone, that dense shell around the inner ear, was the key that unlocked most of these genomes. Hot and humid sites in Egypt, India, and Australia pushed extraction to its absolute limit, which is why some studies rest on a single usable sample. Cold and stable sites, like the Atapuerca caves, preserved DNA hundreds of thousands of years longer. And contamination, the silent saboteur, sat behind the field's worst missteps, from the Mungo mitochondrial claim to the early doubts about Neanderthal ancestry. The master lesson, stated by researcher after researcher, is that genes, language, and culture each move on their own track. A people's ancestry, the words they speak, and the way they see themselves rarely line up as neatly as we expect. And when they fail to line up, the results turn political. Several of these discoveries were controversial, not because of what they found, but because of how they were obtained. The Native American Graves Protection and Repatriation Act, passed in 1990, built the legal framework that shaped the Kennewick Man fight, giving tribes a path to reclaim ancestral remains. The 2011 Aboriginal hair genome became a turning point on consent because the sequencing came before the community consultation. Mungo Man and Kennewick Man were both repatriated in 2017. Two ancient individuals returned to descendant communities in the same year, on opposite sides of the Pacific. Behind these cases sits an uncomfortable archive. Western museums hold vast collections of hair, tissue, and bone gathered during the colonial era, often without anything resembling consent. Each specimen is now a potential genome, and each genome belongs, in some real sense, to living descendants. Out of these disputes grew a new norm, that community consultation should come first, before research begins. Movements for indigenous data sovereignty press the point that genetic information from ancestors is not simply a scientific resource, free for the taking. The tension is genuine, and it does not resolve cleanly. Scientific access promises knowledge that benefits everyone, while descendant rights protect dignity, sovereignty, and the wishes of the dead. Different countries struck different balances. A Danish ethics board approved the hair genome, while Australian and American frameworks placed far more weight on community authority. Eske Villerslev, who worked on several of these studies, shifted visibly over the years toward partnership and shared decision-making. And here the phrase "too controversial to publish" takes on a second meaning. Sometimes a result is held back, not because anyone fears the truth, but because publishing it without consent would itself be the wrong. Restraint, in those cases, is an ethical choice, not censorship. Yet once a result is out, no one controls how it is used. Ancient DNA does not stay in the laboratory. It flows into living arguments about who belongs, who came first, and who has the deeper claim. Cheddar Man became a proxy in debates over British national identity. The Tarim mummies were pulled into the contested history of Xinjiang, where rival narratives fight over who the original inhabitants were. The Egyptian genomes fed straight into the long-running dispute over the race of the pharaohs. The Rakhigarhi skeleton was conscripted into India's Aryan migration culture war, with the same single genome cited as proof by both sides. The Yamnaya story shows the danger at its sharpest. A finding of 90% population replacement in Bronze Age Britain was easily twisted by far-right and white nationalist groups into tales of conquest and racial purity. The irony is steep because the data say the opposite of what those groups want. Every population, examined closely, turns out to be thoroughly mixed, the product of wave after wave of migration. There is no pure lineage anywhere to defend. David Reich made that argument directly, and his 2018 opinion piece on genetics and race still drew a sharp open letter from 67 scholars who worried about how such findings would be framed and misused. This is the gap that makes researchers hesitate. A genome is a probabilistic, hedged, carefully qualified thing. A political slogan is blunt and certain. When the two meet, the qualifications fall away and the slogan survives. Scientists face a real responsibility in how they describe population replacement, admixture, and ancestry, because the words travel further than the data. Three broad positions have emerged. Some argue for full transparency, publishing everything and trusting the public. Some argue for cautious framing, choosing language that resists misuse, and a few argue, in the hardest cases, for withholding. None of these debates would exist at all without the machines that made the genomes readable. Every discovery on this list rests on a technical revolution that arrived in a rush. The first piece was next-generation sequencing, which can read billions of short, broken DNA fragments at once. Ancient DNA never survives in long, clean strands. It comes in tiny, damaged pieces, and only high-throughput machines could reassemble a genome from such wreckage. The second piece was hybridization capture, a method that acts like a magnet for genuine ancient sequences, pulling them out of a sample swamped with modern and microbial contamination. The third piece was discipline. Clean room protocols, bleach treatments, and strict controls kept modern DNA from masquerading as ancient. The fourth was anatomical. Researchers discovered that the petrous bone, the dense case around the inner ear, preserves DNA far better than any other part of the skeleton, which transformed success rates almost overnight. There was even a way to tell real ancient DNA from fakes. Genuine old DNA carries a specific damage pattern, a chemical change called cytosine deamination at the ends of fragments. That signature both authenticates true sequences and, in the Mungo case, helped expose a claim built on damaged DNA mistaken for an ancient lineage. Svante Pääbo tied these threads together and effectively founded the field of paleogenomics, work recognized with the Nobel Prize in 2022. The laboratories of Pääbo, David Reich, and Eske Willerslev grew into something like industrial-scale genome factories, producing hundreds of ancient genomes per study, where once a single sequence was a triumph. Costs fell, throughput soared, and the 2010s became the decade ancient DNA exploded. Limits remain. The hottest, oldest, and most degraded samples still resist, and large parts of the tropical past stay genetically dark. But the reach keeps extending, and each extension reopens a story someone thought was settled. Each of these 10 genomes did the same quiet thing. It rewrote a story that living people thought they owned. A nation's first inhabitant, a desert's mysterious mummies, a civilization's founders, an ancient riverbank skeleton, all turned out to be more tangled, more mixed, and more surprising than the inherited narrative allowed. Ancestry runs deeper and messier than identity wants it to. The dead do not respect modern borders, modern categories, or modern politics. They carry their own histories, and those histories rarely flatter the certainties of the present. There is a pattern in the controversy itself. Every one of these results was once labeled too dangerous, too uncertain, or too provocative to share. And everyone, in time, became ordinary knowledge, folded into textbooks and consumer DNA tests and museum displays. The discomfort faded, and the facts remained. Along the way, the ethics frameworks forged in these disputes over consent, repatriation, and indigenous data sovereignty now guide a whole field, so that the next discovery can be made with more care than the last. The frontier keeps moving. Researchers now pull DNA from cave sediment, with no bones at all, and reconstruct whole populations from the dirt they left behind. They push the recoverable past further and further back, and fill in histories that written records never touched. Through all of it, one finding holds steady. Movement, mixture, and replacement are not the exceptions in the human story. They are the rule. Somewhere right now sits a lock of hair, a hand bone, or a single tooth, still holding a secret that will unsettle something we are certain we understand. The only honest response is to keep asking what else we have gotten wrong about where we came from,